Hello everyone, thanks very much for joining us this evening. Just to say that I'm the Head of Support Services for those of you who don't know me, so please do get in touch with us if there are any questions or anything that comes up, anything you want to talk through, either as a result of this webinar in particular, but anything at all at any time, then do get in touch with us. It's me and my colleagues Laura and Julia. We're available 10 AM to 5 PM Monday to Friday on the support line, email, instant chat, text message, however you'd like to get in contact with us. Hopefully there is a method that will suit your preferences. So we'd really like to hear from you. So please don't hesitate to get in touch with us. As Alice mentioned, do drop your questions into the chat or the Q&A, and after Dr. Hall has done the main part of her talk, then we'll have the Q&A and I will read out those questions, but I won't mention anyone's name. So so you can just drop in questions, but they will be treated confidentially. So I will hand over to Dr. Marcia Hall, and I'm delighted to welcome her. Those of you who've been to some of our members' days back when we could have face-to-face events may have seen Dr. Hall talk before to us, although not on this specific subject, and she's always a really popular speaker, and her talks are always really informative and really enjoyable. So I will hand over to her now.
Thank you very much, everybody. I'm just going to share my screen, but just before I do, what I've done is I've prepared a lecture. It's a little bit medical. It's very difficult with rare cancers to cover absolutely everything, and you'll see why, because there are a whole long list of rare cancers. So what I've done today is given you a little bit of an overview of the direction that the UK Gyne cancer community are taking with respect to rare cancers, which is very much in line with international thoughts as well. How you can help with that comes at the end, but also in the middle I've tried to concentrate a little bit on 2 or 3 specific rare cancers, which are perhaps a little more common. I know it sounds a bit odd, but they are. And at another time, we can perhaps go into some other cancers, particularly if you're asking specific questions about them. So I'm very happy to repeat this, maybe concentrating on other things and also updating you on what we can do. But it is quite a difficult subject. And the last thing I want to say is it's really difficult talking to a computer screen because you really don't get any feedback. So I apologize in advance if I say anything that upsets anyone. I don't mean to do that. Fortunately, I don't know all of you individually and I don't know all your circumstances, but that's obviously where Anna comes in, and I'm very grateful to her for adding that support, because it's always a bit of a worry to us saying all of this when we know some of this information could potentially be quite upsetting. Okay, without further ado, as I say, a little bit of this is fairly medical, but I'm hoping you'll find it interesting. And I will share my screen. Here we go. So let's just get on there. Right. Share. Oops, what's happened to that now? Try again. Hang on. I am screen sharing, right? Good. Okay, so hopefully you can all see my screen. Is that right, Anna?
Yes. Yeah.
Okay, so this is a project. I've called this Talk Rango because there's a project which we started back in 2012. This is the Gynecological Clinical Cancer Group, which is based, or was based, sort of part of the National Cancer Research Institute and the NIHR, National Institute of Health Research. And we, almost 10 years ago, said, look, these ladies with these rare cancers are really not getting a good deal here. We've concentrated so much on common cancers, and we find that every now and then we come across patients who have these rare tumours, and we're really not doing a great deal in terms of research into them or collecting what in fact we are doing with them. So the first step was, as far as we were concerned, to actually first collect some of the information that we could from the UK about how people are currently managing these various rare cancers. And neoplasm is just another word for cancer. So we've called this, one of our pathologists named this rare neoplasms of gynecological origin, RANGO. And thanks to all the people on the left-hand side there who actually have supported this initiative. And one of the reasons for highlighting all of this is that the year before, in 2011, there was a European initiative, and indeed Cancer Research UK and NCRI have an initiative around rare cancers. And an article came out saying rare cancers are not so rare. I think we all understand that. Interestingly, it split up the rare cancers according to the parts of the body, if you like, where they might have occurred. And you will see that the hematological, so those are the leukemias and lymphomas, account for the vast majority of them, 22%. The female genital tract was second, 18%. So that is what we're going to concentrate on today. And you can see why now we're getting so concerned about this, because actually that probably makes up quite a large proportion of these so-called rare cancers. So we do have to be a little bit better at doing this than perhaps we have before, particularly when you look at, say, lung cancers, where you have 8% respiratory tract, prostate and those sorts of cancers, very few are rare, etc. So I think it became more important for us to really sort of look into this in a good way. And the first thing we did, and this is a very long list of lots of medical terms, but the first thing we did was we asked our cancer registry location, which was at the time, it was Trent Cancer Registry, who looked after the gynecological cancers. And we said, could you give us a list of the rare cancers that you have recorded over the last 2 or 3 years? And this is just a very small portion of that list that they produced. And you'll see it's headed ovarian cancer, because when we were talking about rare cancers, we're also talking about uterus cancers, cervix cancers, tubal cancers, anything that is gynecological but rare. And you'll see out of this ovarian cancer section that the classification is somewhat duplicated. So if you look at number 86313 and 86403 and 86703, they all mention Sertoli, Leydig and steroid cell tumours, but they're actually all part and parcel of the same thing. And it's quite difficult, I think, for us to understand from this sort of just straightforward list as to how we're managing these patients and what's becoming of them, because that's obviously the other thing that we need to know. How are we managing them, and are we managing them in a way that actually improves their outcome? So we thought, well, we've got quite a long way to go here. The cancer registries actually have changed since 2012 when we asked for this list, and they probably are a lot more accurate, certainly in terms of the classification, but they still don't really record the treatments that we undertake for these patients, although they might record some of the outcomes. So there's still a little bit of wriggle room that we need to get that information as to how best to manage these. Now, just a little bit of anatomy. This is a cartoon of an ovary with the 2 blood vessels coming in on the right-hand side. And the vast majority of the common, if you like, cancers, although ovarian cancer is not particularly common, is highlighted on the right there with what's called surface epithelium stroma. And that actually is the cancers that arise or come from the outer surface of the ovary and the tube, and those are the list of the cancers that we see from those areas underneath the badly spelt epithelium. But anyway, I can't change that now. What we're talking about today are going to be the rare tumours, which are the ones that occur usually inside the ovaries, and by that I mean there's germ cell tumours, which I'm not going to dwell on today, but we can at another time, and sex-cord stromal tumours, which I am going to talk about a little bit today. Now, obviously, there are other organs as well, and it's a little bit different with the other organs in that the rare tumours arise in the common places, if you like, so it's not quite the same as with the ovary. But if you look at the ovaries in particular, and we'll concentrate on them first, we've already said, looking at the diagram before, that most of them, 60% of them, are going to be of the common subtype, if you like, and those words in red are some of the more common ones, and the serous ovarian cancer is probably the most common. There is, in fact, another type of serous ovarian cancer, which is called a low-grade serous ovarian cancer. And this accounts for about 10% of the 60% epithelial tumours. So it's still only about 6% of the total. Now, low-grade serous ovarian cancer is another one that I'm going to cover in a little bit more detail this evening. So I'll just finish this list and we'll probably come back to that. It is one of our list of rare tumours, and I'll explain why it's the only one that sort of arises potentially from the outside of the ovary. So the other ones, the sex-cord stromal tumours, which we're covering today, Granulosa cell, Sertoli Leydig, which can occur independently, so either Sertoli tumour or a Leydig tumour, but most of them are combined as a Sertoli Leydig tumour. They account for about 2 to 5%. Of ovarian tumours. And then germ cell tumours account for the most, but the majority of these are what are called dermoids. Now, dermoids tend to be benign, and they are really, in effect, the ovary tissue running a little bit amok and just sort of deciding to do things without really any jurisdiction by developing hair and teeth and generally showing that they can form little bits of the body. But they form them in a benign sense, and they really just form a sort of space occupying lump, which causes some problems, usually in a younger age group, but it can occur in older ladies. And it's something which generally the surgeons remove, and we don't have to do any more. There are a very small proportion of those, however, that may turn malignant, and we're going to cover a little bit of that today. Then there are lots of others which I'm not going to cover today, but I can on another occasion. So you can get sarcomas arising from the ovary. You can get these two next ones, which we're going to cover a little bit, the squamous cell carcinoma and the neuroendocrine ovary tumours. They usually arise in the dermoids, because this is where the ovary has decided it's going to show you that it can produce a lining to the lung or the mouth. That lining is called a squamous cell lining, and unfortunately, as it produces that, it develops a malignant potential, and hence it turns into a cancer. And neuroendocrine is usually a malignancy arising in some sort of glandular structure that the ovaries have decided they can show you it can produce without necessarily you asking for it. Stromal ovarii we're not going to cover today, that's to do with thyroid, and borderline tumours we're not going to cover today, but we will talk about them a little bit, you'll see, with the low-grade serous ovary cancers. So let's start with these. So these are similar to high-grade serous only in respect of the fact that they look like clear-looking cells under the microscope. And that's really why they're called serous ovarian cancer. It's a pathological description that the pathologists make when they look under the microscope at these cells. It tends to affect a younger age group, on the average of sort of 40 to 50 years, although the range is quite wide, from 20 to 80 really. Overall, these patients who develop this particular type of ovary cancer have a longer survival if you compare them with the other serous ovarian cancer, high grade. And we think that they probably arise from benign ovarian cysts. And these are called cystadenomas or borderline serous tumours. So they actually are very different to the high-grade serous ovarian cancers, which we now know usually arise from the tube and actually have some sort of precursor changes in the tube before you develop the invasive cancer. On the whole, the low-grade serous ovarian cancers are much lazier, and they're quite slow growing. So they're actually quite slow to present, which means that patients have often been unwell or have experienced some symptoms for quite some time. And unfortunately, this slow growth means it's quite difficult to treat medically. And a lot of my patients will, I don't know if any are dialing in, but a lot of my patients know that I quite often say to them, well, look, if a cancer divides very fast, then our chemotherapies and our drugs and things often work a little bit better. If they divide slowly, because the drugs actually target the division process, it's more difficult to treat. So the treatment of these low-grade serous ovarian cancers is basically surgical. So surgery, and more surgery. So the reason we can undertake this is because it's so slow growing. So when people first present, a good surgical operation to remove all of the low-grade serous ovarian cancer that can be seen is the best start that anyone can have in managing this condition for themselves. Sometimes that then doesn't actually cause any more problems for 5 or 10 years, in which case they may still be fit enough and well enough to have more surgery, and the same thing can happen again. And again, if it's another 5 years or so, they may still be fit enough, and therefore they could have the same sort of procedure again. But that does run out of steam a little bit as people get older. And then it becomes a little bit more challenging to have that sort of surgery, and the recovery takes too long and there are too many risks. At that point, there are one or two other options. First of all, we have explored hormones in this particular cancer, and there's no doubt that estrogen seems to not exactly help this cancer grow, but it certainly, it likes estrogen. So if you give an anti-estrogen, something like tamoxifen or letrozole or anostrozole, which are all anti-estrogen treatments, which are predominantly used for breast cancer patients, but we use them in this type of cancer, then we know that we can stabilize things. We may not necessarily cause a remission, which we would love to do, but some do, about 10-20%. You can get a shrinkage of tumours, but you'll certainly stabilise things for some time. And there was a study published about 4 or 5 years ago now, where a professor in America, Professor Gershenson, followed what he actually looked retrospectively about 90 patients of his who had, some of whom had been started on hormonal therapy as soon as surgery was finished. I'm not sure that's something that I would recommend right now, because if you've had good surgery, you may not need any of the hormones to begin with, and it's quite helpful to keep them up your sleeve. But he noted that those who had hormones actually had probably a year's extra time before they had to have any more treatment. So I think one can extrapolate a little bit from that and say, okay, that's really helpful information, and it's proven to us, if you like, that hormones can be a useful addition. The next thing I want to talk about is Beversomab, or Avastin. Now, Avastin is something that targets blood vessel growth. And just like in high-grade serous ovarian cancer, these low-grade serous cells can travel all around the tummy cavity and spot themselves onto the outer surface of organs and the lining of the tummy cavity. And we know in these tiny little spots that they're very reliant on trying to grow a blood supply from adjacent organs. So if we can try and stop the growth of those blood vessels, then we can challenge that cancer and hopefully stop it growing. Unfortunately, low-grade serous ovarian cancer, because it's rare, we don't have the clinical trials that we have in high-grade serous ovarian cancer, where we can put 1,000 patients in from across the world and say, right, this really works. So we're reliant on small numbers in either small trials or in retrospective reports where patients have, where doctors have said, I have treated X number of patients with this, and this is the outcome. And certainly we do have a number of those small reports. So we've got some mostly from the States, but from Europe as well, with a dozen or so patients in each, suggesting that Avastin, again, like the hormones, doesn't necessarily cause a remission of the cancer. So I think the remission rate was also about 10%. But it does seem to prolong the time before we need to change any treatment. So one paper certainly quoted that the last chemotherapy, because occasionally chemotherapy is used in this, and I'll explain why it's not in a minute, but from the last chemotherapy, they were suggesting that the remission, instead of being 7, 4 and 12 months, was something like 15, 40 and 123 months. So it clearly holds things very nicely, which is really all we can ask for. And as I often say also to my patients, one needs to think about these types of cancer as a chronic disease. It's something with which one has to learn to live, and we have to tinker with the treatments to make sure that that continues with as good a quality of life as possible. So Avastin is certainly something which probably should be explored. In the UK, it's a little more challenging. It's not currently funded, but there probably would be ways around some of that, which I can expand on if you wish. Then finally, there's a group of something called Mech Inhibitors, and in particular a drug called Tremetonib. Now, this all came about because we noticed that these low-grade serous ovarian cancers contained some mutations which are found actually quite commonly in colorectal or bowel cancers. And we know that there are a group of drugs that target patients with these mutations. They haven't actually been found to be particularly effective in most of the bowel cancer patients. There's only one small subset where they're helpful. But we thought we ought to probably look at this in the low-grade serous ovarian cancer population. And the trial done in the UK and the US in parallel was reported last year. And it did show, again, about 9 to 12 months benefit for patients on these Mech inhibitors. On the back of that, the Tremetonib is now funded as part of the Cancer Drugs Fund, which is fantastic news, because it's about time we had something to offer all the ladies with this particular type of cancer. And certainly the first patient that I gave it to was back in June last year, and she remains very well indeed. In fact, she was actually in the hospice because she's had her low-grade serous ovarian cancer since 2008. She's had 3 lots of surgery, and really she was having some symptom control treatment. But really we were running to the end of what I could find for her. So I was delighted to be able to offer her this. And she went home quite quickly and improved dramatically. How long that's going to last for, I don't know, but so far so good, which is wonderful. A word about chemotherapy. So in the past, there have been lots of reports where chemotherapy has been tried in this low-grade serous ovarian cancer. And I think we're becoming better at identifying the low-grade nature of this cancer. So perhaps 20 years ago, the pathologists and some of the surgeons and some of the oncologists, perhaps you wouldn't have recognized some of these patients with ovary cancer as having this particular low-grade type, because it does present in exactly the same way as the high-grade, with the quite profound involvement of a lot of the tummy cavity. And therefore, we probably misdiagnosed some people 20 years ago. So a lot of our reports in the publications are retrospective and rely on reports from 20 odd years ago, which may not be 100% correct. And for this reason, we've got a diverse group of results relating to chemotherapy in ovarian cancer, ranging from a couple of German studies which suggest that 25% respond to chemotherapy with low-grade serous ovarian cancer. If you contrast this to the high-grade serous ovarian cancer patients, where over 90% respond, you can see it's significantly less, but I would challenge them to suggest that even 25% respond. The more recent studies suggest it's more like 5 to 8%. And that's why I think a lot of us are not terribly keen to undertake or to offer chemotherapy to patients who've usually been through pretty major surgery. I think it's probably the last thing that they really need when there's very little chance that it will help. However, there are currently, and we're getting together globally much more now, which I think is an excellent way forwards, because none of us in any one country really have sufficient numbers. And there are some trials of something called CDK4/6 inhibitors. Now, these are a group of chemotherapy-like tablet drugs, actually, which the breast cancer doctors have used now for the last 5 years or so. Interestingly, I did my PhD well over 20 years ago. No, it was probably about 30 years ago on CDK4 and 6. So this is of great interest to me. But they are beginning to use these inhibitors with hormones. And the reason for that is because we have found over the last sort of 10, 20 years that this group of tablet chemotherapy agents work much better with anti-estrogens, which obviously would fit into this low-grade serous model. So that's all I'm going to say about low-grade serous ovary cancer. And we're going to move on a little bit to another group of cancers, which are called sex-cord stromal tumors. My husband just came in and looked over my computer as I was preparing this and said, "What on earth are those?" And it's a difficult name, and I think all one can say again is this is an old-fashioned, rather descriptive name. And if I go back quickly to the picture of the ovary, you'll see there that the ovary has its blood vessels. It has these little germ cells, which are the little eggs that are inside the ovary, and there's only one pictured there, well, there's two, I suppose. And then around the egg, there'll be some development as the menstrual cycle goes through, but we all have to have some scaffolding around all of these bits and pieces, and that scaffolding is called stroma. And it's the stroma where these cancers develop, and that's why they're called sex cord stromal cells, because they're from around what we call the sex cords, which is where all of these little eggs, and in men who also have sex cords, obviously it'll be the development of sperm. So that's why they're called what they are, and it perhaps is just a descriptive term, but we know of two common types of sex cord stromal tumours. The first is much more common, if you like, even though it's rare, than the other one. So granulosa cell tumours are much more of the common type of sex cord stromal tumour. 50% of them actually occur under the age of 50. But again, there's quite a big range from 25 to 80, and there is also a juvenile form of sex cord stromal tumour, which is much rarer, but it does exist. I don't know a great deal about it, not being a paediatrician. Fortunately, the granulosa cell tumours, the vast majority of people will present with stage 1A. And if you have a look at the cartoon on the left-hand side, you can see there what I mean by that is that it's just confined to the ovary and it's within that capsule, the epithelial surrounding. That cartoon actually depicts all sorts of cancers, which is why you've got stage B and stage 1C there. 1C is where you've got a bit of cancer on the outside surface, but it's very unusual for granulosa cell tumours to do that. Occasionally they can be quite large, and if they're very large, they can, under some pressure, burst, and they can also sometimes twist on their vascular pedicle. And by vascular pedicle, I mean that little strip of tissue that comes out to the purple blob of the ovary, which contains the blood vessels. And we might be able to understand that if you develop a lot of weight in one side of the ovary, it may twist, and that then cuts off the blood supply, which is rather a painful thing to happen. But that can also then lead to a bursting of it, and that's where we may run into trouble with recurrences from this type of cancer. So the vast majority present with stage 1A, confined to the ovary. If you look at the middle panel here, it's quite distinctive when you look at them under the microscope. And what this middle panel depicts is something that the pathologists call cal-exner bodies. And they're supposed to look a bit like coffee beans, and they do actually. So you can see all of those little coffee beans there. And that is a picture of what these cells look like under the microscope. Unfortunately, although most present with stage 1A, about 25% of those will have this granulosa cell tumour come back again. The vast majority are after really quite a long period of time. And we're talking here about 20% of them even over 20 years. And it's something which I think we need to discuss as a gynecological community, hopefully with patients, because it's something that comes to us in the clinic as a bit of a challenge. So you meet a patient first of all after their surgery, and you've got good news, it's only a stage 1A, you've got a 75% chance nothing else is going to happen, but you've got this chance something might happen, but it probably won't happen for a long period of time. So the options are that we either keep you on some sort of follow-up, which sounds a bit boring to me if you're going to have to, it's probably much easier now we've had COVID and we can do telephone consultations, but it used to be coming in to see us really just to be told that you're absolutely fine and off you go again. Or we explain it all to you nicely and then explain that you can contact us if there are any problems and let you go away. And if you're concerned about something, or you think you might need an ultrasound just because you're worried that there may be a lump in your tummy again, or if it's 20 or 30 years later and I and the other doctors have all retired, you can at least mention it to some other people that you may see. That may be another way of dealing with this. And I think some of these rare tumours, one of the things that we've got to explore in some of our research is how we approach things like this, where we know there is a real chance this could come back, but really it's an awful long time, and we don't want to worry people unnecessarily. We don't want to investigate them unnecessarily, and equally we want to have our resources, I suppose, just to treat people that really need it at that time, if you like. So that's something that we'll come back to in a minute. The other thing about these particular granulosa cell tumours, and actually this applies to all of the sex cord stromal tumours, is that they can secrete hormones. And the most common hormone for the granulosa cell tumours to secrete is estrogen. So, and we're coming on to treatments in a minute, and you'll see that some of the treatments are also anti-estrogens. So on the right here is an MRI scan of, with the white arrows, you can see a sort of lumpy looking thing, and that actually is the granulosa cell tumour. And on the left, you'll see a black looking blob with two little white blobs inside it joined with a line. And that black looking blob is in fact a cross section of that lady's uterus. And what often happens with about 40% of the ladies who have these granulosa cell tumours is that the estrogen secreted from the tumour itself, which is depicted, as I say, with the white arrows, actually affects the lining of the uterus. And it can, and I can tell you now that this lady who is postmenopausal, so she's finished all of her periods, those little white blobs shouldn't be there at her age. They could be there in a 20 and 30-year-old, but they certainly shouldn't be there in the 67-year-old lady that this is. So we know that the estrogen from that tumour is causing activity in the lining of her womb. In other words, it's sending messages to that lining saying, please prepare, there may be a pregnancy on its way, you may need to look after an egg, etc., etc. And that unfortunately can also lead rise to womb cancers. So we do, in about 10% of cases with granulosa cell tumour patients, also find an early uterus cancer. And for that reason, we usually recommend that we don't only take the granulosa cell tumour out, but that we take the uterus out and the other ovary as well. Having said that, if people are at the younger age of the granulosa cell tumour, and it's less common, but if they are, and they still wish to pursue families and things, then there will be options for fertility sparing surgery. Just we would have to keep a close eye and probably sample the uterus before we decide we can definitely do that. So moving on to the other group of sex called stromal tumours, they're a group called Sertoli-Leydig cell tumours. They're more rare. They're definitely unilateral. Granulosa cell tumours occasionally are bilateral. Most of the Sertoli-Leydig cell tumours do present quite young, so in the second and third decades, and only under 10% of them are over the age of 50. Now, 50%, it's said, have signs of excess male hormones or androgens. So they may have excess hair in places they don't want it. They may have a deeper voice. They may actually be balding with a sort of male pattern baldness. I have to say, I've never been particularly struck in the patients that I've seen with Sertoli-Leydig tumours about this androgenic excess, but I have certainly, I certainly recognise that it can occur. And it probably occurs more in patients who have more advanced Sertoli-Leydig cell tumours, which will be even rarer than most of them. Theoretically, just like the granulosa cell tumours, they can also come back very late if they present with a stage 1A, again, 20, 30 years later. Theoretically, they can come back again. It's something which I think they're a rare tumour. I think the chance of any one of us individuals seeing it is probably pretty small. So in terms of treatment, both of these, again, surgery and more surgery, I'm afraid, but certainly granulosa cell tumours can be what we call debulked. It's a terrible term, but certainly we can remove that if they're fit and well. Now, I want to tell you a little story about the literature because it highlights to you how difficult sometimes it is looking back at historical information. So in terms of the treatment of these two tumours, I've mentioned to you that hormones can be useful, certainly for granulosa cell tumours. They're probably less useful for Sertoli-Leydig, and I'll explain why in a minute. But in granulosa cell tumours, we recognise that anti-estrogens such as tamoxifen and letrozole can cause some remissions and can certainly stabilise these cancers and stop them getting any worse. And in the literature, if you looked at the reports that were published there, it would suggest that up to 70% of ladies with advanced granulosa cell tumours who aren't suitable for any more surgery or perhaps are waiting for things to grow, make the surgery worthwhile, are treated with hormones, and 70% will respond. In other words, the cancer will get smaller. So when our Australian colleagues actually said to us, look, we'd like to do a proper study of anastrozole in granulosa cell tumours and other gyne cancers, we jumped at this and said, yes, that would be great, because we would really like to look at this properly. And anastrozole is one of those anti-estrogens that, like I mentioned. And I think that the one thing we've always been worried about retrospective studies is that they tend to be somewhat anecdotal. And by this, I mean that it really depends on what you're told. And you can see from the cartoon here, 90% expressed satisfaction, but actually 10% just didn't make a comment. And I think as doctors, we certainly have an inclination to remembering either the really, really bad things or mostly the good things that have happened. So when you're reporting retrospectively, you often think about the patients that have done very well. And you might well therefore have some sort of bias in reporting them, which is why prospective studies are so useful, because you're collecting patients as you go, and you therefore can't miss out either the very bad ones or the mediocre ones or whatever it happens to be. There's no bias to it. So when we did this study, we finished this study about 5 years ago. And again, just to give you a little bit of an idea how difficult all of this was, it was open in a lot of the Commonwealth countries, so Canada, Australia, the UK. And we, over 3 years, managed to accrue 41 patients, so not very many. But out of those 41 patients, all treated with anastrozole, precisely 10% had a response. So very different to the 70% that's reported in the literature, and obviously a much more accurate outcome. Having said that, that's actually people where the cancer shrank. There were far more people who, again, were stabilized on the anastrozole. So this is not going to stop us using hormones, because if we can achieve some stability for patients, fantastic. Until something else comes along where we can take it away or shrink it, that is better than nothing. So certainly that's not going to put us off, but it just gives us a much better idea and much better information with which to discuss patients' treatment options. And these anti-estrogens don't come with no side effects, although they are probably better tolerated than many other treatments. So the other thing about granulosa cell tumours that we've discovered recently is that the vast majority of them have a mutation in a gene called FOXL2. They can also have inhibin and calretinin seen when you look at them. And we're hoping maybe this discovery about FOXL2 will lead to some novel treatments. And certainly you'll see later on when I've finished the talk that there are some plans to look at how we might collect groups of patients who might benefit from studies that look at novel agents and new drugs. And the other thing about granulosa cell tumours is they can sometimes be associated with other familial conditions, such as something called Peutz-Jeghers syndrome. They're quite rare, but they're certainly something that one needs to bear in mind. Just turning briefly to the Sertoli-Leydig cell tumours, these tend not to be quite so hormonally responsive or treated so well. In fact, the majority of them, as we've said before with the granulosa cell tumours, are also stage 1. And when you look at them, they're what we term well differentiated. That simply means, again, that they're lazy and they look like the cells they're supposed to look like, which is always good news when cancers come along, because if they look horrible and like other cells, then they probably are pretty horrible. There are some aggressive subtypes. These are obviously even more rare, and those ones do tend to be treated with chemotherapy, and they certainly do have a poorer outlook. And just as in granulosa cell tumours, we have found a specific mutation, but not in all of Sertoli-Leydig cell tumours, probably only in about 50–60%. And this is something called a DICER1 mutation, which we may be able to use at some stage in the future for new treatments. Okay, so getting back to my original story, which is that in 2012 we thought, okay, we've really got to do something with this. We initially explored what other countries were doing, and in Australia they've got a system called Cartwheel, where they've got patients actually reporting their own rare tumours. We felt we would rather have doctors do it if we could, or medical teams, because there are lots of little bits of information that we probably would value having, which patients may not know. We requested support from the CI UK and NIHR because of their rare cancer initiatives, and then we're told, well, that yes, we have got these initiatives, but there's no funding available. So that was that door. We hadn't got much further forward apart from discussing things to do with biobanks and creating tissue and looking at research in these rare tumours. So when another pathologist came on to the committee, we thought, right, we just have to begin writing the protocol and see what we can get going. So we began writing the protocol, and this is all part of the bureaucracy that goes into undertaking a proper formal clinical trial, which we felt was the best way of trying to approach this problem. And we then applied to a charity called the CTRT, which is actually a local charity of mine, and they very kindly gave us some money for a data manager for the next 3 or 4 years. And Target Ovarian Cancer also gave us a small amount of money for a laptop, which has been helpful. And my trust, the Mount Vernon Cancer Centre Trust, which is East and North Hertfordshire, actually agreed to sponsor the study, which is the other thing we need. There's no money involved, but it's certainly something which is required to get going with all of these things. Now, while all of that was going on, here's a nice pretty picture which I've adopted as part of the logo for Rango. And this requires quite a lot of explanation, so apologies if it goes a little bit over the top, but if you have a look down the left-hand side, you'll see a list of different types of cancer, and we'll just concentrate again, well, there are two really we can concentrate on. One is the Ovarian Adenocarcinoma, which is the fourth one down on the left-hand side, a nice blue colour, and the second bottom up is endometrial, which is a sort of mid-red colour. And if you look for those two colours on the wheel, you'll see that the endometrial cancer one is actually at the sort of 7 o'clock position, and the ovary cancer one is almost opposite at the 1 o'clock position. Now, what this cartoon or this clock face, if you like, predicts or actually shows us is that this is the genetic information taken from a group of cancers which were collected in something called the Cancer Genome Atlas. So patients donated their tissue, DNA was extracted from the tissue, and then the entire genome was sequenced. And we were trying to find out what's the basis of these cancers. Is there a simple genetic code that codes for these cancers? And the answer is probably no. But certainly if you look at the ovary and the uterus cancer ones, there's a large blue and red hump, which represents the vast majority of them. So some of them are very similar, and those will be the more common ones. You will also see, however, that there's a kaleidoscope of colour coming out of the sort of towards 8 o'clock position for the uterus cancer or womb cancer ones, and towards the sort of 3 o'clock position on the ovary cancer ones. And what this represents is the similarities of cases of ovary cancer in their DNA that actually match with other cancers. So if you take the ovary cancer one as an example, you'll see there's a nice blue splurge that links very nicely with something called BRCA, which in fact stands for breast cancer. And this is probably represented by a group of patients who have a BRCA or breast cancer related gene, and they are known to be at risk of ovary cancer as well. So that probably accounts for that link. But you will also see in both the uterus and the ovary cancers that there are strings leading to leukemias, leading to colon cancers, leading to lung cancers, leading to thyroid cancers, leading to brain cancers. And this gives us an idea that the genetic makeup of some of the cancers that are classified as ovary or uterus cancer actually genetically resemble different cancers altogether. And it makes it even more important that we try and identify the groups of people with those cancers so that we can treat them hopefully better. So we started the summary, and initially we kept it very simple, and we said, look, what we would like to do is actually collect information about patients who have these rare tumours. We can't really include absolutely every rare tumour. You saw just part of that list, but we would actually specify certain rare cancers to begin with. And what we want to do is to understand what's currently happening. So how are our colleagues treating these patients up and down the country? We'd like to know, if we can, what's actually happening biologically. And for that, we probably need access to patients' tissue. And I don't mean that all you lovely patients have to go off and have biopsies. I mean that if you've had some sort of surgery, which the vast majority of people with these types of cancer have had, then that specimen will still be in the pathology laboratories. People can't understand that, but it is. And we would like access to small portions of that so that we can actually study them in the laboratories to see whether there's new things like the mutations that I've just discussed, FOXL2, DICER1, which may be able to lead us to a greater understanding of how these diseases occur, why they occur, and potentially what sort of treatments might be available. And interestingly, in the past, when we've looked at some rare tumours, sometimes that's actually then been applicable to the more common cancers as well. So we set ourselves this target of just saying, okay, can we collect permission from ladies to amalgamate their clinical information and permission to collect some samples? And we have these things called endpoints when we set up a trial, and we wanted to obtain tumour samples and also blood samples. And the blood samples came along for a different reason, which I might go into because it's somewhat amusing, but only to do with bureaucracy and how life is really. And the secondary endpoints were to do with ensuring that the clinical details that we collected, and everything we collect is anonymous, but we need to make sure that it does align with the cancer registries. And part of the reason for this was that the new English cancer registry called Encore got slightly miffed that we were doing this study, and so they said to us, well, we're collecting all of this data. And when we said, well, are you collecting what treatment is being given to these patients, they said they had to admit that they weren't. But it would be very interesting to compare what we collect with what they collect, just to see how accurate their collection is, which really relies very much on records rather than sort of informed clinical teams, I suppose. We also wanted to just compile the accurate confirmation of the incidence and outcomes, and the treatments and adverse effects, and finally a register of the location of the tissue samples that were available. And the reason for this is because we had tried to explore something called a tissue bank, and there's an awful lot of bureaucracy and legislation around setting up tissue banks, and we thought, well, where are we going to put it? Added to which the local hospitals sometimes don't want to let go of their tissue. Some of that is because for medico-legal reasons they need to keep it for a while, which I quite understand. Some of it's because it's a very small biopsy, so there are some very rare tumours, particularly of the cervix, which I can go into on another occasion, where we literally have a very small needle sample and we don't have anything else. So it is important, therefore, that some of that sample is retained at the local hospital. So we wanted to set up something that was easy to use for doctors and yet would have the patient's obviously interest at heart. And what we thought we would do is run two systems in parallel. One was, if you like, a virtual tissue bank. So where are the tissue samples? And the way that would work is that if a laboratory came along and said, I would like to collect all of the tissue samples of a stage 4 granulosa cell tumours, for example, we would be able to look through our database and say, well, patient numbers XYZ from these hospitals have had tissue taken and there is permission to use that tissue. So these are the contact details, and we will forward their consent form so that you can access the tissue. At the same time, the gynecological pathologists, who are actually, they're internationally really very well respected, the UK British gynae pathologists, and I attended one of their annual meetings a couple of years ago to discuss this study, and I was absolutely amazed at the international configuration of the people that attended from absolutely everywhere, and they very kindly have hired a section of the Barts tissue bank. So there are some hospitals where they say, look, there's so much tissue here and I'm looking at it right now, I can send you a sample right now. So if they want to do that, then they can do that as well. And then some tertiary endpoints, we need to ultimately secure some more permanent funding for this entire resource, which obviously I'm trying to do, and I've got some more bids into CRUK at the moment. We would like to extend the current specified list, and I'll come on to what all of that is. And finally, we would like to intercalate some sort of treatment arm for the study of these various rare gynae cancers. So this is the list at the moment, and you'll see that there's quite a few ovary cancers there. There's something called the Adnexa. The Adnexa is just the portion of the pelvis where the ovaries sit, and embryologically we have what's called the Wolffian origin sort of bit of tissue that comes down as we're forming in utero, and you do sometimes get some funny cancers just of that Wolffian origin, which is what these fat woes are. I don't usually harm people very much, to be honest. And then we've got a few uterus rare cancers, quite a few cervix ones, and then you may be surprised to see vulval cancer down there, but there are only about 1,000 cases of vulval cancer in the UK in any one year. And we really struggle to know what to do with the more advanced ones. So you'll see on this list that we've actually been a little bit specific about what we're going to collect. So if we take vulva as an example, we know that ladies with a stage 1B vulval cancer usually have just a skin lesion on the vulva, which is excised, and very often they have quite a reasonable outcome from all of that. And so as far as we're concerned, that's very good treatment. But there are some patients where either that comes back or they present with a much more advanced disease than just this local skin lesion. And it's those ones that we find very difficult to know what to do with and have much poorer outcomes. So it's those ones that we're interested in collecting. And the same premise applies to further up the list, something called serous borderline tumours, but also mucinous ovary cancer. So there are quite a lot of ladies who present, often quite young actually, with stage 1 mucinous cancer. And the vast majority of those actually have a very good outlook. But those who have a more advanced mucinous cancer tend not to have such a good outlook. And therefore, those are the ones that we actually are more interested in collecting so that we can actually document all of this. So by 2017, we'd actually managed to get all of our health research agency and ethics applications approved. And the last little bit of the jigsaw was actually to do something called portfolio adoption. And this is where I'm just going to tell you a little quick story about blood samples. So portfolio as adoption is because in the UK, we're extremely lucky in that the government does offer some central funding for health research. Most European countries certainly don't have that, and the USA doesn't either. So we're extremely lucky to have that. It's quite tight, as is most of the NHS, and therefore the way that we actually ensure that the support gets to the right research is by having what we call portfolio adoption. So you put your trial forward to be adopted onto the national research portfolio. And if it is adopted, then you will have access to the research nurses that exist in most hospitals. Unfortunately, the portfolio adoption was refused twice. And the reason it was refused is because they viewed this very much as a database collection. And indeed, probably at the time that we were doing that, it was, although we were recording where the tissue was and we were recording details about patients anonymously, etc., they said, well, you're not actually doing any research. And we thought, well, no, we're not. So at that point, I have a little study running for common ovarian cancers locally with our local university at Brunel. And I was collecting some blood samples from these patients. So I said, well, perhaps if we collect some blood samples from these patients, we can assess something called circulating cancer cells, which probably won't exist very much in a lot of these slower, rarer tumours, but they probably will in one or two of the very active ovarian rare tumours and uterus and cervix. So they said, yes, that's fine, that we can adopt it on the portfolio on that basis. So that's how we managed to get it adopted, and it's still adopted. And currently, I think the end date for the study is 2027, but my view is that this should probably go on for longer than that, so that we get a really nice long time to actually collect as many patients and information as possible. And the samples, we send these out to the sites and they're just the various tubes and bags and things that we ask them to collect. And we only take them at the same time as patients would have blood tests anyway, so they're not an extra blood test. And they're usually taken hopefully at diagnosis, but often they've had surgery already. That's fine, because that's obviously made the diagnosis. So we have them before any other treatment, such as hormones or chemotherapy, halfway through any treatment, at the end of treatment, and then maybe 3 or 6 monthly at follow-up, depending on the cancer type. And then the last bit that we had to put in was a good database. Now, these often are very expensive, but fortunately there's an international system that the Australians put us onto. And this is an initiative run by the University of Vanderbilt in the US, which I think is a charitable part of their setup, have set up this web-based database system where we can actually make the forms that we need to fill in, absolutely tailor them to the Rango project, and we can also allow hospitals to put in their own data and to own their own data and simply share with us the anonymized data. Ultimately, we'll be able to interface this with national and international collaborators, and a lot of them are using the same REDCap system. So that will all help ultimately when we actually, hopefully, will put some of the UK data together with some of the European and US and Australian data to see whether we can get bigger numbers and actually identify more of the ways to try and help in these areas. We're collecting information, obviously, about follow-up, any treatments that have been given, if any further operations have been done, if the cancer's come back, hopefully not, and obviously if the patient has died, we would like to know. A lot of the time that may be of something completely different. So it's just really just to get a full picture of things. Now, again, the data protection and all of these things require us to have specific consent. So we actually now do have consent. We have to ask permission to collect dates of death, and also to correlate the patient details with the National Cancer Registry. So we ask that on the consent form for this study. Most patients are perfectly happy with that, but obviously it's something we have to specifically ask. Now, I'm sorry, the map is slightly out of date, and my lovely trials coordinator pulled this together, and I haven't managed to get hold of her over Easter to ask her how she did it and alter the map. You'll see there that a lot of the southwest is open to Rango. There are a couple of yellow centres there, but actually they're now green. Manchester is now open, Leeds and Leicester are open. I don't think Gwyneth and Bangor is open, Belfast is open, Glasgow is open. So there are a lot more here that are open than actually listed. And in fact, altogether, we've got 27 sites open, and we're continuing to roll this out throughout the UK. And last time we looked at the database, there were well over 150 patients. So it's coming along very nicely. There are 44 gynae cancer centres across the whole of the UK. I would like to open all 44. It may be that we open a few more because there are quite a few locations where the gynae cancer centre does the surgery, but you go somewhere else for your other treatments or for your follow-up. So we may have to open a different site as well, but we will keep rolling out for as long as we need to. And then finally, the next initiative, which I'm beginning to work on, is to try and integrate a treatment component to this. So you'll see from this sort of diagram, what we like to do is that at the moment, the patients, the trials teams contact us with specific anonymized patient information, and we say, yes, that's great, we can register the patient. We usually say we would like, or we don't need bloods for that patient. If there's no treatment available, then we will simply register them and follow them in the Rango study alone, and that's what we're currently doing. What we would like to do is to build up a little portfolio of treatment options. Now, there may only be 2 or 3, but it would be nice to have 2 or 3 treatment trials, and we've called these TRANGO, which is treatment for rare neoplasms of gynecological origin. And if we note that the patient that's being submitted to us has a certain condition which we have a treatment trial for, we would then send information to that local research team so that they can consider whether that patient is suitable. And from there, if the patient doesn't want to or is not suitable, they come straight back into the Rango study. If they are suitable, then we can commence the screening investigations. And as I say, this is really a medical slide, and lots of doctors know that it's all very well saying this, but actually the hospitals need to undertake all sorts of checks and balances to make sure that they can do that particular piece of research in that study before they will allow you to put a patient onto a study. And what I'm working with the National Cancer Research Institute is to actually allow us to do that in a shorter time frame. It usually takes 2 or 3 months, and we'd like to do it in a couple of weeks, which I think could be possible, because a lot of hospitals do not want to open studies where you're only going to have 1 or 2 recruits, and with rare tumours that is possibly all you are ever going to have. So it's going to be quite important that we can move fast and only in the centres that actually really need to have it open for various patients. So that's what we're hoping to happen, and work in progress. So that will probably take another 10 years. No, hopefully it'll take less than that. But obviously that's what we're trying to do. Motto for Rango: never give up. And you'll be pleased to hear that sometimes we do get the cheese. And I'd like to thank the whole project development team. Now, I know there've been some questions, and I'm very happy to answer some of those. And I'm equally happy to email people if there are any questions later on that we can't address. So back to Anna and Alice. Thank you.
Thanks very much, Marcia. We have come to the end of our time, so I know that you've kindly offered to answer questions via email that Alice and I will then circulate afterwards. So I think we'll do that. I will just have a very there is one we could answer just quickly, which was the slide that had the list of the different subtypes. The third box was covered by the logo, and someone's asked what it says, so we could answer that one quickly, and then the rest we will follow up with.
Hang on 1 minute. It is so these are the non-granulosa cell sex cord stromal tumors. There's also in there the hypercalcemic small cell ovary cancer, which is a slightly different tumor to, let me just see if I can't move it actually. Yeah, so it's the second group, the Sertoli Leydig cell tumors that I was talking about, and the small cell ovary cancers.
Thank you very much.
I'm happy, you know, that can be circulated with the talk or whatever, so that's fine.
That's great. So so that's what we'll do, and thank you very much to everyone who submitted a question, and you will get an answer to it, but what we'll do is circulate our follow-up email afterwards, and we'll include the answers to those questions in the follow-up email. So thank you very much, and if you do think of any other questions in the meantime, then please do email support@ovacome.org.uk, and we'll forward those questions on as well. But thank you very much to Dr. Marcia Hall.
Just make one other point, and that is, I do need your help, because actually if you could publicise this locally when you next see your doctors at your local centre, make sure they're getting it open or have got it open, and I'm very happy to help, then that would be wonderful.